All right. Well, thank you everybody for joining. I'd like to introduce Dr. Tingwei Mu from Case Western University. He's doing a lot of exciting research in the field of GABA-A receptor disorders. So we're thrilled to have him here, and I'll let him take it away.
All right. So it's really my pleasure to be here with GABA-A Alliance. So I really want to thank Justin for organizing this really nice foundation. This will just provide really nice hub to connect the patients with the clinicians and with the basic scientists. So our goal is to try to find to solve determine the molecular mechanism underlying this GABA-A receptor variants, as well to find out therapeutic strategy to treat the GABA-A variants. Okay, today let me see. Okay. Let me share my screen. So Justin, so I can share my screen, right?
I think I have everybody enabled for screen sharing.
Okay.
I can add you as the host also, if that helps.
Okay, I think I can share my screen. All right. So today I would like to share our recent understanding of this GABA-A receptor variants. So previously, Justin gave me some really good questions about how what's the foundation is really interested in, and what we could do to meet the demand from this from Justin and this GABA-A alliance. So basically Justin gave me 4 questions. This is shown on the slide A. How alpha-1 beta-2 gamma-2 variants are different in your experiments? Are some more responsible to certain mechanism or treatment? B, is there a way to group variants together with each subunit as responding to method number 1, 2, 3, or combination? Number 3 is about the gain and versus loss of function relating to the experiments. Number D is there's the expectation that single agents may modify the disease, or is a focus mostly on working toward the combinations. So okay. So basically by reading Justin question, I can redirect the about 4 questions to the following 3 categories. The first one is about the GABA receptor and their clinical variants. The second is about the disease-causing mechanism of GABA-A variants. The third one is probably what we are really working hard toward is to find therapeutic strategies to correct the function of pathogenic GABA-A receptor variants. Let's start with the first one, GABA receptor and their clinical variants. So probably you already know there's many variants occur in the GABA receptor subunits. Then some of the variants in this GABA receptor genes are pathogenic. So if we go to the NIH ClinVar database, it gives us a pretty comprehensive list of what variants are available in the literature. For example, if we go to ClinVar and input this GABA-A1, this corresponding to the alpha-1 subunits of GABA receptors, we will come out this page. Here we can find that for there's currently about over 200 missense variants in this GABA-A variants, and also you can find that some of the variants has been annotated as pathogenic or likely pathogenic. But we can also find that there's over 100 variants that has not been annotated because their have the uncertain significance according to our current knowledge. This is the most positive because that there's no experimental data to show whether there's some functional defects or not for this missense variants. Therefore, for this clinical variants, there's an urgent need to carry out this variants for their function. But what we could do for now is that we can do some prediction. Before going into the detail about how we can predict the pathogenic probability of such variants, we can have an overview about the currently known variants in the major subunits of GABA receptors. Here we summarize the variants in the major subunits of GABA receptors, namely alpha-1, beta-2, beta-3, and the gamma-2 subunits. We can find that for each major subunit in GABA receptors, there's over 200 missense mutation in each case here. Right? If you read each row, this will sum up over 200 clinical variants. But there's the majority of the variant has not been comprehensively characterized. This means that in the current Tingwei database, is annotated as uncertain clinical significance. But we can also find some of these variants are annotated as pathogenic. So if you have done some of the search about the GABA receptors, probably you will already know that GABA receptors, they're assembled as from 5 subunits. This will form this pentameric structures. This is also where the GABA receptor will function on the plasma membrane. So we can find that the most majority subtype for GABA receptors are composed of 2 alpha-1, 2 beta, and 1 gamma-2 subunits. So in this, you will also find that there will be a central pole coming in the middle. This will allow chloride to move inside once the channel is activated. If we move rotate 90 degrees of this receptor away from us, this will give us another standard view of this GABA receptor. This is supposed to be a functional state. But if some of these variants occur, that will, in some cases, lead to loss of function of these GABA receptors. From here, we also summarize the currently known clinical variants in the primary priority sequence of GABA receptors. For example, on the top is
could you scroll down on the presentation so we can see the screen?
This one?
Yeah, it's still on the main page.
How about now? Can you see this primary sequence of the GABA receptor variants?
It's just showing like the title page.
Title page.
That's all it's showing right now. Yeah.
So let me see.
Oh, there we go. Yep, we're on the ClinVar one. Yep.
Oh, okay. Now it's a ClinVar database, right?
Yep. Okay.
Let me try to see whether still good. Still in this with the yellow, red, and green.
Yes.
Highlights.
Yep. Okay.
All right. So from here, this will give us some idea about the spatial distribution of these GABA receptor variants in the major subunits of GABA receptors. For example, from here, the GABA receptor, each subunit are composed from really long N-terminal domain, 4 transmembrane helices. We highlight the red region as a pathogenic, currently no pathogenic one. The yellow highlights mean that the currently they are known in the ClinVar as uncertain. The green highlights means they are most possibly benign variants, indicates if that variant occurs, most possibly this will have limited effect on GABA receptor function. So we can find that there's some hotspot for this clinical variants that is denoted as pathogenic. For example, the variants in the transmembrane domain 1, transmembrane domain 2, or transmembrane domain 3, they enrich many of the pathogenic variants. surely there's many of these unknown variants in this transplant domain. So it appears to us that the variants in these three transplant domains, they are most likely leading cause some pathogenic phenotype. But we did not show here, but many of the variants in TM4 or the long loop between TM3 and TM4, they are most likely benign variants. Another really interesting hotspot is that that result in the connection loop between different secondary structures. That means that either they're connecting different transplant domains. For example, there's a loop connecting TM2 and TM3. We can find that this is a hotspot for pathogenic variants. Another hotspot for pathogenic variant is in this loop 9. This is the linking between this beta sheet 8 and the beta sheet 9. Let's see. Okay. And also, because there's so many variants had not been annotated in the Tingwei, so we use a pathogenic predictions algorithm to find out whether the variant is pathogenic or not. From here, we use two different prediction software. One is from the AlphaV Sense, which utilize recently developed AlphaFold structures, and also using evolutionary conservation. It will give us really nice prediction about every possible variant in the every possible location in this GABA-A receptors. This will give us some really nice clue about whether a variant can be pathogenic or benign. Here it shows that all these possible over 4,000 variant in the alpha-1 subunit. Let's see. So this is just a really overwhelming information. But if you really want to find about the influence of a variant, how you can do it on your own laptop or on your own computer. So here I just put a tutorial about how you will be able to do it on your own if you are really interested. From here you can find I did a tutorial. You can go to that's the first step. You can go to this open access publication here. You click on this by archive publication. If you go here, sorry. Let me just do a demonstration here. Okay, after you paste your link into this any of the web browser, you will go to this website. From here, you click on supplementary material because all this data are really huge. It cannot be fitted into the main text. You will find there's 4 supplemental tables. Supplemental table S1 is related to the alpha-1 subunit. This is related to beta-2, beta-3, and the gamma-2 subunits. For example, if we want to study the beta-2 variants, we click this download this Excel file. You are downloaded to here, then double click your downloaded file. This will just open the file using Microsoft Excel. So this file is pretty big. Okay, after opening this file, you will find this many of the columns. I will point you to the most important columns. Basically, this column H is the annotation from the NIH Tingwei. Then column C is a prediction using the AlphaV Sense software. Column E is a prediction by the repository software. So we can so this is whether the clinical significance, whether they are pathogenic or benign are the most important parameters you want to focus. For example, if we have a look at this the ATH variant. So in clinical in Tingwei is still gives uncertain significance because probably it hasn't been verified experimentally for its function. But both prediction so will predict this variant will be pathogenic. So most likely this variant will lead to the functional defects for this GABA-A variant. It's also for example for another variant is uncertain significance according to Tingwei, but it's most likely pathogenic because it's both software predict it's gives functional defects. Okay. Let's go back to the PowerPoint. This basically this PowerPoint summarizes the steps. Go to the website, click to download this Excel file. Then this three columns, this column C, E, and H will give you the either the annotation from Tingwei or the predict software. This will give you a first clue about the variant, whether they are potentially benign or pathogenic. Okay. Then next we need to think about the what is the potential disease causing mechanism for this GABA-A receptor variants. I think this is important because after we get to know about the disease causing mechanism, we can talk about the potential therapeutic strategy for each different variant, because each different variant will have different disease or potential different causing mechanism. Before going that, I want to introduce you a little bit about this protein manufacturing factory inside the in cells, because for this GABA receptors, their functional states are assembled as a 5-subunit or as pentamers. Only after assembly of these pentameric receptors, they will be able to traffic from the inside the cell and to the plasma membrane. Only after that reaches the plasma membrane, they will be able to function as a chloride channel. So for each individual subunit, for example, whether it's alpha-1, beta-2, or gamma-2, this is their topology. It has a really large N-terminal domain for transmembrane helices. Each subunit then need to fold into their native or near native states. Then they will be able to assemble with adjacent subunit to form these functional receptors. You can imagine that if something goes wrong with either the folding step or assemble step, this receptor will be retained inside the cell. Then the retained subunits cannot go to their functional states. Then in most cases, they will be degraded really fast by the cellular protein quality control machinery being transported into the cellular degradation machinery, for example, either by the proteasome or by the lysosome. Then during the last 10 to 20 years, our group and other groups, including Dr. Khan's group and Vanderbilt, we begin to realize that actually there's several disease-causing mechanisms, and some of the mechanisms are more common in the GABA-A variants. So I can go through this different class of variants with you. For the class 1 is called the proteostasis defect. So it means this variant will cause some of the folding, assemble, degradation, aggregation, trafficking, and cytosis defects. If we go to this cellular trafficking pathway, as we just mentioned briefly, this GABA receptors they need to be folded in the inside this organelle called the endoplasmic reticulum. Then after proper folding, this GABA receptor will be able to assemble into these 5 subunits states that will traffic from the endoplasmic reticulum through this Golgi apparatus and onward to their plasma membrane. If something goes wrong along this protein transport process from inside cell to the cell membrane, there will not be sufficient amount of receptors to be present on the membrane, thus causing their functional defect.
Dr. Milk, can you put that diagram on the screen, the illustration for that?
The illustration, let me try again. So I did,
yep.
But let's see.
Now we see it.
Okay, now you see it. Okay, all right, not good. So I repeat. So for this class 1 defects, they are caused by their inefficient trafficking from the inside the cell to the plasma membrane, you can find that there's a several potential steps that can go wrong, either by the protein folding step in the endoplasmic reticulum, or their assemble step into this 5 subunits assembly, and their trafficking from this along the way from the endoplasmic reticulum through the Golgi to the plasma membrane. Surely, the second class of defects come from the channel electrophysiologic defects. This can result from the defect in this agonist binding. In the case of GABA receptor, it will be related to the defective GABA binding or channel gating effects. This means that once the ion channel reaches the plasma membrane, there be some like the current deficiency, whether it's caused by this current kinetics or some of the like reduced single channel conductance. Overall, this will be reflected into in this electrophysiologic defects. The class 3 are more like in their very early protein synthesis pathway than occur in the nucleus, because some, for example, if the mutation or variants belong to nonsense or frame shift variants, this will introduce the pro-mutual truncation of the whole protein. Then this will cause the so-called nonsense-mediated MI decay. In that case, the MI level will be decreased. This will be reflected in a significantly reduced protein level. This will also cause functional defects because there will not be sufficient protein synthesized at the same place. All the other variants will be categorized as the class 4 other variants. Okay, so after we get to know about the functional defects, we will be able to find out what is the strategy we can use to correct the different functional variants. So this is before going into the therapeutic strategy that we are working on, I want to give you some examples in the alpha-1 subunits about how these variants might lead to different deficiency. For alpha-1 subunits, currently there's over 200 missense variants. So we select 8 variants in our study. We selected some of the representative variants based on their special distribution. Some of the variants are located in the N-terminal domain. Some of the variants are located in the TM1, TM2, or TM3. And also we select the variants according to their different let me see whether, probably you cannot see my slide, but maybe this one you will be able to see my slide, hopefully. So we can also see that we also select the variants according to their different disease severity, at least according to the prediction. Some of the variants cause a relatively moderate deficiency, some of them more severe defect. Okay. Then we studied the functional deficiency for all the select 8 variants. This is from the patch clamping electrophysiology recording using HEC293T cells. HEC293T cells is the most commonly used expression system for ion channel proteins, including GABA receptors. These are human cell lines, so they are more related to the human physiology. So from here, figure 8 displays peak current amplitude for Y-type receptor and all these variant containing GABA receptors. The black curve is the response from the Y-type GABA receptors. All the 8 variants displaced reduced current amplitude compared to Y-type. Surely some variants are more severe compared to some other variants. What is interesting that the most severe variants occurred in the transmembrane domain, as displayed by these 2 curves.
Slide up 13. Is that the one you're looking at? I think that was the one.
Slide
right?
13, yes, slide 13, yes, slide 13. Let me just insert slide number so we will be able to. Okay, this is slide 13. Then we can find that according to the peak current amplitude, all the age selective variant lead to loss of their loss of function, and we continue to determine the EC50 values for these variants and the Y type. We can find that on figure B, Y type is let's see, Y type is this one, the black line here, and also this Y type here. We can find that all these variants are either have equal potency for GABA or some reduced potency for GABA. This is also consistent with that all these variants lead to loss of function of GABA receptors, at least for this age selective variants in the alpha-1 subunit. Okay, so this is now slide 14. So the GABA receptor can have loss of function due to different mechanism, as we just mentioned, either due to their loss of trafficking to the cell membrane or loss of their gating properties. Then we use the Western blot to determine the total protein expression level as well as the surface protein expression level for these 8 variants. It's very clear that the majority of this variant will lead to reduce the total protein level as well as the surface expression level among these 8 variants. Surely, this one exception is called this T289P variant. This variant did not reduce their surface expression, and this variant also the location for this variant is in the TM2 region in this alpha-1 subunit. So it's also interesting that many of these TM2 region cause gating defects. This is probably because the TM2 occupies most inner part for this for that import. Therefore, any variant that occurs in the innermost part of the channel will likely influence their gating properties. Okay, now we're going back to slide 14. So from here, we can find that the majority of these selective variants will lead to their reduced surface trafficking. This will also, in addition, we did some of the surface staining, confocal immunofluorescence staining to visualize the surface expression of the variants. For example, this is one example. This is the A323 variant in the alpha-1 subunit. In Y-type cases, the red staining means the surface receptor, indicating that the receptor can traffic to the surface efficiently. This is now slide 15.
Advance the slides for us so we can see them. I think we're
Can you
stuck
see?
on 13.
Okay.
There we go.
Nice. Okay, now we can I need to go back to this PowerPoint presentation mode. Okay, now slide 15. We can find that the red staining represents the cell surface GABA receptors. In the Y-type cases, we can find really extensive surface staining. This is indicates that GABA receptors are effectively trafficked to the cell surface. However, in the case of variant A323 variant in alpha-1, essentially we could not find any of this surface staining because this variant is very cause a really profound misfolding of the whole subunit. Therefore, that will be degraded really fast inside the cell. That will not be able to track it to the membrane at all. So this will be also reflected in a significant reduced current amplitude for this GABA receptor variant. All right. So for us, since there are many of these variants will lead to their loss of surface expression, we try to find a way that can restore their surface trafficking as a novel therapeutic strategy to crack GABA receptor function. So this is important because so now it's slide 16. Okay, because if many of the current drugs on the market, if they are target GABA receptor, they will work on this GABA receptor only after they reach the plant membrane. If as a first place, GABA receptor cannot traffic or cannot reach their final destination for function, then the current drugs will not be able to have some influence. Therefore, we try to work on this protein quality control or the protein folding trafficking machinery inside the cells. DNA is transferred into RNA and it translated into protein. Proteins that need to form their polyfolded states, right? But there's some missing step which we can label it as a black box, right? During the black box, protein will form their functional states. We try for many of the GABA receptor variants that have deficiency during this protein folding step. We will work on this protein folding step as a way to create their folding trafficking and that cell function. So in the lab, we mainly focus on using two different strategies. One strategy is called so-called protein-specific regulated small molecules. For this kind of small molecules, they are general protein folding enhancers. So this class of small molecule, they will be able to promote the protein folding, thus to promote the protein assemble and enable their trafficking from the inside the cell and to the plasma membrane. The final goal is that the correct misfolding will be reflected into an enhanced protein trafficking to the membrane for their function. So remember, this class small molecules, they are not GABA receptor specific. They will have a general role for the cellular signaling pathway. So the second class of small molecules we focus on is called the pharmacological chaperones. This class of small molecules, they are GABA receptor specific, meaning that these small molecules will bind to the GABA receptor directly. The binding behavior will stabilize the otherwise misfolded GABA receptors. This will enable their further assemble and traffic to the plasma membrane for their proper function. So you can find that both small molecules are promising because for proteostasis regulators, this will target the general folding pathway that will likely work on different subunits as well as different misfolding prone disease. Pharmacological chaperones, they are specific, meaning that they could be developed tailored to this GABA receptor subunits, depending on the mutation, the location of the variant. And also, a nice way to think about the two class small molecules is that they have different mechanism action. Therefore, the potential combination use of these two classes of small molecules will be able to produce a synergistic or additive effect to rescue some of the really severe GABA receptor variants. Okay, so now we are advancing to slide 18. So in the lab, we did some screening effort to discover the potential pharmacological chaperones for GABA receptor variants. So it's very fortunate that because the pharmacology of GABA receptor have been studied very extensively. So these 5 subunits, people know this GABA or the agonist binding site reside between 2 subunits, alpha and beta. And there's also very famous class of drugs called benzodiazepine that bind between at the interface between alpha-1 and the gamma-2 subunits. Then we screened the currently known GABA receptor modulators for different variants of the GABA receptor. In the case, we focus on the alpha-1 variants, including the DN, GD, and PL variant. From this initial screening, we found that there's several very effective compound in increasing total protein expression level for these GABA variants, including hispidolin TP003. In some cases, like this CL compound, CL218872, as well as this midazolam, are also effective in increasing the GABA receptor from protein expression level. It's very interesting that all these effective compounds belong to the positive allosteric modulator for these GABA receptors. We can have another look at this, these two most effective compounds. We are now at slide 20.
Could you just stop on, we missed 19, could you stop on that for a second so we can see it?
All right, so now it's, can you see slide 20?
Yeah, I can see 20. Can you just stop? Can you just go to 19 real quick? Because that one didn't show up on the screen.
Okay. Okay. So this is slide 19. So this demonstrates a binding site for agonist and then benzodiazepines here. So from here, we found that the effective compound is producing TP003, in some cases, midazolam and Cl218872. All these four compounds belong to the positive allosteric modulators of GABA receptors. Then we can go to slide number 20. Okay, for slide number 20, this gives the chemical structures histidine and TP003. These two compounds that both cross a blood barrier. I mean, this is probably the common case for many of the GABA receptor modulators. So we did as a dose response for these two chemicals. We can find they have the micromolar ECV values for a number of different variants. Then we continue to determine whether the compound application will be able to increase the surface expression level for this GABA receptor variant. In this case, we tested two variants in the alpha-1, GD-PL variant in the alpha-1, and also we use patch clamping recording to determine whether drug application will be able to increase their GABA-induced current, because you can think that even if only we can increase their cell expression, but its receptor is not functional, that probably will not be beneficial for this variant containing GABA receptors. Indeed, we demonstrate that application of these pharmacological chaperones can not only increase their surface expression level, and also they will be able to create their function deficiency according to the peak amplitudes. Up until now, in most cases, we use human HEC2 T cells as a model cell line system to perform our research. But probably you're also wondering, because GABA receptors are residing in the central nervous system, probably the most relevant expression system will be the neuron, right? So in order to determine the effect of this pharmacological chaperones on GABAergic neurons in the central nervous system, we develop this use genome editing to knock in this disease variant into apparent healthy iPSC cells. After the generation of these iPSC cells carrying this disease-associated variant, we differentiate this iPS cell into GABAergic neurons using different differentiation approaches. After differentiation into this GABAergic neuron, we use imaging techniques to determine whether drug application will be able to enhance their surface expression. So shown here as a result for this one variant called this G251D variant in the alpha-1, we can find that application of histidine TP003 will be able to enhance their surface staining for this variant. The quantification of this variant, this result are shown in Figure C. Okay, so now we are really pretty confident that the application of the pharmacological chaperones will be able to enhance the. Now we are at slide 20 and slide 21. We are confident that the application of this pharmacological chaperone will be able to increase their surface expression as well as to correct their functional deficiency once they reaches the plasma membrane. Okay. So in the lab, now we are advancing to slide 23. In the lab, during the last 10 years, we also identified several classes of proteostasis regulators that have similar effect in increasing function of this GABA receptor carrying the receptors. This includes the activators of the folded protein response, calcium channel regulators, as well as some of the FDA-approved drugs such as dinaprost and dihydroergocristine. If we advance to slide number 24, so basically we will give you an example about how we identify such small molecules as a proteostasis regulated. Basically, we express GABA receptor in the cell culture system when we screen this approved drug library and use chemiluminescent-based assay to quantify its GABA receptor expression level. From that high-throughput screening, we identified at least two compounds that are effective in increasing the protein expression level for different GABA receptor variants. In this case, dinoprost and dihydroergocristine. We both DNP and DHEC that cross the blood barrier. DNP is prescribed to induce labor. DHEC is used to reduce blood pressure and treat dementia in some cases. So indeed, we collaborate with Dr. Angelo Caramidas at Queensland in Australia. He did some really nice patch clamping recording for us. Basically, what he demonstrated that both DNP and DHEC, they are pretty selective for this variant containing GABA receptors. They can increase the peak current for this DM mutation in alpha-1, RG mutation in gamma-2, RQ mutation in gamma-2. This is summarized in this quantification figure, but these two compounds don't have a lot of influence on the wild-type GABA receptors. I think this is also important because we want to achieve selectivity for the variant compared to the wild-type, because we want to minimize the potential of target effect or the potential side effect for this kind of proteostasis regulators. Okay. So now we advance to slide number 27. Okay, so this is basically the summary slide. So I hope today I demonstrate you about the following three take-home message. First, reduce the surface trafficking and the loss of function of GABA receptors is a major disease-causing mechanism for GABA receptor variants. The second is restoring proteostasis pharmacologically corrects the surface expression and function of pathogenic GABA receptors, representing a promising therapeutic strategy to treat GABA receptor variant-related genetic epilepsy. The final take-home message is that several lead compounds such as histidine and dihydroergocristine were identified as effective reagents to correct the function of GABA receptor variants. Their further development is needed for translational application. We are just working hard for this translational application for these small molecules. So finally, I really want to thank my group member who did all this hard work. So we have a really talented team. Basically, many of this work I shown today was done by Dr. Megan Wang, and also by Dr. Meng Wang, Dr. Xiaojing here. And also we have some really serious, serious collaborators. For example, Dr. Jeff Kelly, Dr. Luke Weissman at Scripps, Dr. Jin Chung Kang at Vanderbilt. We collaborate with Dr. Carol Midas in Australia. We also have some collaboration with Dr. Raj Nashmi, Ashley Sheffer, Dr. Chris Richards. Some of the work were not shown today. Finally, I want to thank the funding of the NIH, which makes this work possible. Okay. All right. So also, I want to thank you for your attention. If you have any question, I will be happy to answer. Thank you.
All right, Dr. Mu, thank you. It was very, very detailed, and I really enjoy hearing about all the stuff you're doing. And if anybody has any questions at all, we definitely have some time. If not, I have a few that were submitted that I can ask, but I'll give everybody a chance to unmute or just to type in the chat if you'd like.
Very easy, fine with me. Probably you can ask, so I can answer directly.
So I'll probably start with the ones I have here. Are there drugs where you're seeing a differing activity in the A1, B2, and G2, such as this TP003 seem to be more active in G2 subunits versus A1 or vice versa?
If our current understanding is that as long as the variant is has a folding deficiency. This folding enhancers and the pharmaceutical chaperone, they will have some kind of efficiency. Surely, whether a compound is more effective or the other compound is more effective, it really depends on the location of the variant. If we think about the successful stories from the CFTR field, in that CFTR field, they found different characters that target different location of the variant. If the variant is close to the binding site for these characters, they are more effective, right? So this is will give us a really nice clue for GABA receptors. I think in short that this pharmaceutical chaperone and processing regulated, they are more or less will be effective. But in order to see some of the really severe variants, probably a combination of two chemicals will be the best choice.
I guess you may have kind of addressed this elsewhere in the presentation, but do you find that the variants causing mild, moderate, and severe disease all seem to have the potential to benefit from the process that you're using for correction?
Right. So both mild variants and the severe variant, they should be able to benefit from this kind of small molecule approaches, right? So it's also about the final whether this activity can be restored to the wild type level. For mild mutation, probably the correction will be more efficient because for mild mutation, they will keep some of the residual activity for GABA receptor. Therefore, if we can increase the activities a little bit more, then probably reach the clinical significant level. But for severe variants, right, so probably that will need more effort, maybe a combination of two, that to reach the near wild type level.
Okay, and then the last question I had was, do you find there's any variants, or I guess specifically like a frame shift mutation or other kind of unique variants that would be harder to treat or present challenges or kind of be like in their own category? I know you mentioned, I think some of them will be like class 3 and 4. Do you find those are harder to treat?
For this, for class 3, like the nonsense and the frame shift mutation, because that will cause premature truncation of the whole protein. So am I understanding that it will be more challenging to be corrected? But unfortunately for GABA receptors, most of the probably all the patients, as far as I know, have the heterozygous variants, meaning that the patient will carry one white allele, one the variant allele. So probably for the nonsense and the frame shift variants, it's critical to enhance the activities of remaining white allele. That will give us some pretty nice therapeutic strategies. As long as this remaining white allele activity can be enhanced to a significant level, this most so I will speculate that it will lead to clinical beneficial.
Great, thank you for that. Let's give everybody else a second if anybody has a question. All right, so I guess we can wrap up. I really appreciate your time and see all the work you're doing. It's it seemed like a few years ago there was a lot less going on, and it seems like you've made some progress, and we're all excited to see where this goes, and glad to help any way we can. So, thank you a lot for your time.
Yes, thank you, Dr. Mew.